Okay, great. So thank you everybody for tuning in. I guess first of all, I hope everyone and their families are okay. I hope that you guys are stocked up, that you're doing as well as we can in this time of crisis. And I hope that this will briefly sort of take your minds away from what's unfolding outside our doors. So first of all, it's my great pleasure to introduce you to today, Joe McGirr, the first graduate student to graduate from my lab 5 years ago today. And Joe has been with me since the very beginning of my career at UNC. And I want to just show you this here. Let's see. Sharing this. Okay. See if this works here.
I can hear on YouTube, but you can only hear me through Zoom. No, I can hear on Zoom.
So let me just mute this here. That's not going to work. So Joe wrote me 4 days after I started my faculty job at UNC. and from the very beginning, I said that he was not allowed to join my lab. And yet here he is. So, and let me just check, make sure everybody can still hear everything I'm saying. I think so. Okay, great. Let's try this again. So I should say up front, Joe, that I won't have the funds for you to settle in my lab. And yet he did so well. Oh, right. And so notice, this is July 9th. Then I hear nothing from Joe until October 8th. This is 5 days after my first daughter was born. And my response, I am completely swamped with a new daughter right now. New daughter this week. Sure enough, he writes back and everything works out. And in fact, Joe did so well on his first rotation project that it was published as his first chapter. In that December, it was accepted in Molecular Biology and Evolution. So going from March to December with his first paper published is pretty remarkable. Joe joined the lab. It's not beautiful Caribbean reefs like you guys are imagining. It's these desert sort of salt panish lakes where the pupfish live. Here he is hauling buckets. And Joe is also fantastic in the field. In fact, that very first trip, his luggage didn't show up. And me being so excited to get into the field, we ended up bushwhacking through all these thorns. And before I realized it, Joe was completely torn up. No complaints whatsoever as we went to get these pupfish. And this is kind of what the fieldwork looks like. Here's Joe and Emily and Michelle. And probably the most memorable time was Joe and Emily and I going to this remote island, Rum Cay, in the Bahamas, which I don't even think Joe will talk about today. But we ended up getting dropped off by this guy with 40 dogs in the middle of nowhere and set on the wrong path. So we were lost basically the whole day bushwhacking. And Emily and I are kind of reserving our energy and preparing for this long hike. And Joe is frolicking like a gazelle the entire time. So here I am hauling buckets and pipes. Here's Joe taking a picture. Here I am again, Emily and I hiking through this underbrush. There's Joe, happy as a deer the entire day. Joe's second chapter, which you won't hear about today, what actually happened with this, it was actually scooped a few months earlier by the only other grad student working on San Salvador Island pupfishes. But the amazing thing is that Joe ended up publishing in a better journal because he came up with a comparison that he'll talk about today that Ezra did not come up with. And in fact, Joe then took Ezra's data and showed that the same signal was there of parallel expression. And I think that's pretty fantastic. And then for his next two chapters, he took the lab in a completely new direction into allele-specific expression. And let's see, here we are collecting in the field in North Carolina. And there were also some ups and downs. So this is the point where Joe learned to fear me. So there was actually an alligator right in this pond when we first arrived. And Joe realized I wanted to catch the alligator with the lab. So just notice how close he is to the edge of that pond. by that alligator that's still in there. But we did collect pupfish that day. This was probably one of the hardest things about grad school, was that Joe had an entire project that he won't talk about today trying to get CRISPR to work. And instead, the sort of whole committee advised him to go in this direction of allele-specific expression. But so Joe, I want to sort of show these pictures to show just how diverse Joe's skill set really is. He's made what I hope are lifelong friends in the lab, which is, it's really unusual. I mean, Joe is he's a really nice guy, genuinely nice guy. He's easy to get along with. He's sort of efficient, productive, intelligent in all the ways that you could hope for in a grad student. And it's been sort of one of the luckiest experiences of my career having Joe to join the lab, particularly when he had, so many fantastic choices of Dana Matute, Bob Duronio, who have also served as his mentors. And now he's joined Chris Willett's lab in these last year as my lab moved across the country. So here's Joe after passing his quals with lots of pupfishes everywhere. And I'm so sorry we can't be there today to celebrate. And this is also just an example of how sweet Joe is. He actually gave me this art that he made himself of a pupfish right as he joined the lab. I mean, who does that? So I'm extremely fortunate to have had Joe in my lab for these last 5 years. This has been sort of the most rewarding experience of my career, is mentoring grad students, in starting with Joe and watching him sort of develop into a full-fledged scientist now ready to take on the world. So with that, I think that covers everything. I'll give it away to you, Joe. Let's see.
All right, let's see if this works. Thank you, Chris, for that really kind introduction. I don't know if anyone else was able to hear it. I really hope so, but if not, I heard it and meant a lot. So thank you very much. And thank all of you for being here today, tuning in to my remote thesis defense. This isn't exactly how I imagined I would be giving my final talk as a grad student, but I'm really grateful for the opportunity to present all my research with you nonetheless. Chris, if there's any audio issue, can you call me and I'll try to make adjustments? If not, I'm just gonna get right into it. All right, so today I want to share with you an example of what I think is one of the most captivating things about the process of adaptation, when species can evolve the ability to extract new resources from their environment. I study 3 rapidly diverged species of Cyprinodon pupfishes, and I want you to pay special attention to differences in their faces, because these differences tell a story about how each of these species are uniquely adapted to a different ecological niche. Now, this story involves rapid speciation and ecological divergence, 2 hallmarks of adaptive radiation, which you might associate with classic examples like Darwin's finches or East African cichlids, where in both cases, a common ancestor rapidly gave rise to numerous species exhibiting divergent craniofacial morphologies that are adapted to these different feeding strategies. For example, we know that the big beaks on these finches are great for crushing large seeds, and the downturned jaws on this species of cichlids are really great at scraping algae off of rocks. And these types of awesome matches between diet and morphology are really widespread across adaptive radiations around the world, and are thought to result from ecological speciation, a process where divergent selection favors divergent adaptive phenotypes and reproductive isolation evolves between populations. Now, this process unites principles of both evolution and ecology, and so research in these systems has been central to our understanding of mechanisms driving species divergence. However, even within the most famous and well-studied systems, we're only beginning to identify genetic changes that give rise to different adaptive phenotypes and maintain species boundaries. To address this, my research examines adaptive radiation from a genetic perspective, using genomics and transcriptomics to examine adaptive morphology, parallel evolution, and mechanisms of reproductive isolation. And this research is really motivated by long-standing questions in evolutionary biology, including how does genetic variation translate to phenotypic variation? How predictable is evolution? And how do interactions between genetic variants contribute to reproductive isolation? So today I'd like to show you my study system and explain why it's great for asking these types of questions. And then I'll move on to my thesis research split into 3 parts. First, I want to explore how nature builds new traits by asking what are the types of genetic changes and changes in gene expression that affect adaptive morphology. And then I'll show you similar genetic changes between species that you might not expect just based on their morphology. And finally, I'll show some evidence for a genetic mechanism that's potentially limiting gene flow between species by asking how selection shapes patterns of gene expression in hybrids. All right, so first off, I told you that adaptive radiations make great study systems because they link evolution and ecology. But why do I study this adaptive radiation in particular? Well, like Chris showed, these fish live on some really pretty islands in the Caribbean. My lab and I, we go down and we sample in these shallow hypersaline lakes that are on the interior of these islands. And we're looking for species like this, Cyprinodon variegatus. This species has a really wide range from the coast of the United States down through the Caribbean to the tip of South America. And throughout this range, pupfishes are usually dietary generalists, like this guy, meaning that they mainly eat macroalgae, detritus, and small invertebrates. However, there are 2 very interesting exceptions to this rule on the small island of San Salvador, which is only about 11 miles long and covered in these shallow hypersaline lakes. Here, there's been a recent adaptive radiation where we find the generalist pupfish alongside 2 endemic specialist pupfishes. And I'd like to talk about each of those species. First off, I want to talk about Cyprinodon bronitoroides. I'll refer to it as the molluscivore. This species has evolved much shorter and more robust jaws relative to the generalist species, as well as this novel nasal protrusion that it uses as a wedge to pry snails out of their shells. And here's a quick video of that in action, where our molluscivore is using his little nose to pop his meal out of its shell in the cutest way possible. So that's our molluscivore. And then the other specialist species, Cyprinodon desquamator, which I'll refer to as the scale eater, has alternatively evolved these really long jaws, as well as this more elongated body that allow it to remove scales from other fish in quick strike attacks. And so I'll refer to this species as the scale eater. Here's that in action, some slow motion footage of a scale eater male going after a generalist who just gets out of the way. And notice the dark coloration of this male scale eater. This is his breeding coloration, and he's in his breeding territory. And I just want to point out that the scale eaters get much darker in their breeding coloration than either of the other 2 species. And so that's our cast of characters for the day, the San Salvador Island pupfish radiation. And there are several things about this system that make it very strange, that also make it great as a genetic model for ecological speciation. First off, if we go back to our textbook examples of adaptive radiations, where species have diverged hundreds of thousands to millions of years ago, these pupfishes have diverged much more rapidly within the last 10,000 years. And yet we still see, like these other systems, divergence in craniofacial morphology. Another interesting thing about this system that I mentioned is that these 2 specialist species, the molluscivore and the scale eater, are endemic to just a single island in the Caribbean, despite many other islands in the Caribbean with lakes that have similar ecological characteristics to the lakes on San Salvador. And we've searched a lot of different islands. I've searched some myself, like this remote island of Mayaguana where about 300 people live. I've also searched less remote places like the Exumas. Here I searched a lake that's right behind a liquor store and across the street from where the infamous fire festival is supposed to take place. But my point here is that even in this lake, it looked similar to lakes you'd see on San Salvador Island, but no specialists. The only species I found were the generalist species. And this is true of a lot of different islands we've looked at. So what's special about San Salvador Island is that we find these two specialist species living alongside the generalist in many of these semi-isolated lake populations. And this is interesting because it suggests that there's some barrier to reproduction between species that are maintaining these species boundaries. And we see that there is significant pre-mating isolation between species. And what I mean by that is if we were snorkeling and looking down from above at fishes, we might see a male courting a female of his same species. And that accounts for most matings that we see in the wild. However, we do sometimes see interspecific mating between species. And when we bring these species back into the lab, we can cross them in all directions and they produce fertile hybrids. And so when we create F1 hybrids, they often show craniofacial phenotypes that are intermediate between parental types. So usually F1 hybrids look kind of like generalist species. So together, this makes a great genetic model for ecological speciation because these species have rapidly diverged, are in sympatry, and can still hybridize. But despite their young age, we see dramatic differences in adaptive phenotypes, including divergent craniofacial morphology. And for the rest of the talk, I'll show generalists as red, the molluscivore as green, and the scale eater as blue. And so this brings me to my first part of my research, asking, can we identify genetic changes between species that are important for differences in those novel traits that we see in the specialists? So for this question, I had 58 whole genomes. Most of these genomes were derived from individuals from San Salvador Island. I also had some generalist outgroup populations from throughout the Caribbean, as well as a couple outgroups from Mexico and Death Valley in California. And then I used a standard variant calling pipeline to identify over 13.8 million SNPs among individuals. And most of those individuals, like I said, came from San Salvador Island. So one of my first questions was, out of these millions of SNPs across the genome, are any of those unique to either of the specialist species? And so to get at this question, I measured FST, which is a relative measure of genetic differentiation that ranges between 0 and 1, where 1 indicates that a SNP or an allele is unique to a population, also termed fixed between populations. So looking at these three different pairwise comparisons, looking between the molluscivore and the scale eater, which are the most diverged species, found that there were over 6,000 SNPs fixed between them. Fewer between the generalist and the scale eater, about 1,500, and just 79 fixed between the generalist and the molluscivore. And this was a surprising finding. We did not expect so few SNPs to be fixed between species showing such drastic divergence in complex morphologies. And to put this level of genetic differentiation into perspective, we can look to bird genomes, where between golden and blue-winged warblers, there are about 74 SNPs fixed, and just 82 between hooded and carrion crows. But my main point here is that the primary difference between these species pairs is plumage color, pigmentation differences. My results are suggesting that a similar low number of SNPs are fixed between species showing dramatic craniofacial divergence. And so if my question is, can I identify regions of the genome associated with differences in this craniofacial morphology and other adaptive traits, it might be a good idea to start looking at the regions of the genome containing this fixed variation. And so here's an example of that, where we have two genomic regions containing SNPs that are fixed between the molluscivore and the scale eater specialists. And this gene is unannotated for any gene. This region is unannotated for any genes, and this region contains a gene called GALR2. Next, I looked across each species genome for evidence of hard selective sweeps, where selection favored a single haplotype to rise quickly to high frequency in a population. So if we imagine a hypothetical ancestral population of chromosomes represented by these lines, and these empty circles represent neutral variation, or SNPs, then one day we have a beneficial allele pop up on this genetic background. Over time, recombination and selection might shape a population of chromosomes that look like this, where our beneficial allele has swept to fixation in the population, dragging along with it this neutral variation and shifting the site frequency spectrum towards high and low frequency derived variants. So looking for this pattern across each species genome, I found that these two regions containing fixed variation also showed evidence for a hard selective sweep in the scale eater populations. I also looked at patterns of within population nucleotide diversity, so for each species, as well as between population absolute nucleotide divergence to verify that these regions showed signatures consistent with selection and not confounding processes like low recombination rates in these regions. So finally, I performed genome-wide association mapping to ask, can we find regions of the genome that are associated with variation in jaw size between species? And so I used two different approaches, one Bayesian and one frequentist. And I found that there were a lot of regions of the genome that were implicated as associated with jaw size by both strategies. Here is what the output of the Bayesian strategy looks like, where we have the black and gray alternating bars representing our 9,000 scaffolds that make up our reference genome. And these outlier points here are 20 kb windows that showed the strongest association with jaw size variation between species. So taking my best candidates from the genome-wide association mapping and overlapping them with my best candidates showing signs of selection, I found that there were a total of 31 regions that contained 18 novel candidate genes never previously associated with craniofacial phenotypes, and 11 genes that had been annotated for effects on skeletal system development from other model systems. And so this was really exciting. And one of the exciting things about this region in particular, containing the gene GALR2, is that it falls within a previously identified QTL explaining up to 15% of jaw size variation between species. Another interesting thing about this region is that out of all these SNPs here that are fixed between species, none of them fell within the protein coding region of the gene. And so that suggested that the next piece of this puzzle was to ask, do these variants that are fixed between species influence differences in adaptive traits by changing gene expression levels between species? So to get at this question, we go back to San Salvador, where now I'm sampling all three species, but I'm sampling total mRNA, which is a way to look at genes being expressed in each species. And I had two different lake populations for each species. So here's what I did. I set up purebred breeding tanks. And so this was set up with the same species from the same lake population. We collected their eggs and let them develop for 8 days. At this point, I extracted whole larvae RNA and we sent out for sequencing. And then we also let some individuals develop for 20 days. And at this stage, they're a little bit bigger, and they're big enough that I was able to dissect craniofacial tissues and extract RNA directly from those tissues. So then I generated read counts and used a standard gene expression R package, DESeq2, to identify genes that were differentially expressed between the generalist species and each of the specialist species at both of those developmental time points. And so these red points indicate that genes are differentially expressed. Each one of these red points is a gene that's differentially expressed in each of these comparisons. And I found that the most expression divergence was between the generalist and scale eater at 8 days old. Now, excitingly, overlapping my candidate genes that were significantly associated with jaw size and showed signs of selection, I found that many of them, 11 of them, were also differentially expressed between species in at least one of these comparisons, including these genes in bold here that were known to influence skeletal system from other systems. And so there's a lot of information here, and there's a lot of really cool stuff that I want to get into with this same data set. But first I want to pause and summarize what this data tells us about the genetic basis of adaptive morphology in this system. So first off, we see that there are few variants that are fixed between species, and that some of these variants seem to be associated with divergence in craniofacial morphology and are near genes that are known to influence skeletal development. And then the gene expression data shows us that many of those candidate genes that are under selection and show signs of being associated with jaw size are also differentially expressed between species, suggesting that fixed variation may influence divergence in craniofacial morphology by regulating differences in gene expression between species. Okay, so now coming back to this data set, I want to talk about this because there's an interesting pattern here that only reveals itself when you look at the intersection of comparisons. I want to talk about parallel evolution of gene expression in this system. So we know from many examples in nature that when species are faced with similar selective pressures, they often respond with the same adaptive solutions. So a lot of times when we think about parallel evolution, we think about things like color or morphology, but we can also think about parallel evolution at the genetic level, asking whether similar phenotypes are evolving through similar changes in gene expression or all the way down to the nucleotide level. And studying these systems can teach us a lot about evolutionary constraints, as well as potentially help us to predict evolutionary trajectories. And while there's been a lot of research on parallel changes at the genetic level underlying parallel changes between similar niche specialists, there's been little research on parallel genetic changes underlying divergent specialization. And that's why I want to talk about this system. So if we look at this 8-day-old data in a more simplified way, I found that there were about 6,000 genes differentially expressed between the generalist and the scale eater, and about 1,000 differentially expressed between the generalist and the molluscivore. Looking at the overlap of these comparisons, I found that there are a bunch of genes that seem to be uniquely differentially expressed in the scale eater comparison, and just a sliver that are uniquely differentially expressed in the molluscivore comparison. But this overlap are a bunch of genes that show differential expression in both of the specialist species relative to the generalist. And this was immediately surprising because 80% of genes differentially expressed between the generalist and molluscivore were also differentially expressed between the generalist and the scale eaters. Now we wanted to know about the direction of differential expression of these genes because it can tell us something about selection acting on gene expression. So if we were to randomly draw a gene out of that overlap, out of these 818 genes, it might show the same direction of differential expression in specialists where it's upregulated in both specialists relative to the generalist or both downregulated, or it could show opposite directions of expression where it shows high expression in one specialist and low expression in another. Now, a neutral model of gene expression evolution might suggest that out of these roughly 800 genes, we might find about 400 showing each direction of differential expression. But what we actually find is this. Almost every single gene that's differentially expressed in both specialists relative to the generalist was differentially expressed in the same direction. And that means if I were to take randomly a gene out of this overlap and it was upregulated in scale eaters, chances are very likely it's also upregulated in the molluscivores. And this is really exciting because it's one of the first examples of a weird pattern. We see significant parallel evolution of expression between divergent trophic specialist species. So this is weird because I've told you all the way that these specialist species are divergent from one another in their craniofacial morphology, in various behaviors, in their male breeding coloration. And it really begs the question, what do these specialist species have in common that might arise from similar changes in gene expression relative to the generalist species? And so there's one maybe straightforward answer here. Maybe the specialists are more related to one another than either is to the generalist species. However, if we look at our phylogenetic tree of all of our genomic samples, it looks like the scale eaters form one group, and then the generalist and molluscivore tend to group by lake population. And so this would suggest that the parallel evolution of gene expression that we see is not due to a more recent split between specialists. Another answer might come from gene ontology enrichment analyses. So looking at those genes from the overlap that showed differential expression in both specialists, we found that they were highly enriched for metabolic processes, whereas genes showing unique patterns of differential expression that was unique to each of the specialist species, we also found enrichment for metabolic processes, but this is where we started to see enrichment for things like cranial development and pigment biosynthesis, more obvious phenotypic differences that we see between the specialist species. And so together, this might start to paint a picture where parallel changes in gene expression might be a parallel adaptation to higher trophic levels in the specialists, where scale eating and molluscivore niches and their more nitrogen-rich diets present similar metabolic requirements relative to the lower trophic level of the generalists. And so this is data from a previous study that looked at stable isotopes from dissected muscle tissue that was looking for relative differences in trophic position between species. And we can see that the scale eater occupies the higher trophic position with the most nitrogen-rich diet, followed by the molluscivore, and then at the lowest trophic position are the generalists. And so together, this might suggest that, well, first off, we're showing to our knowledge the first evidence of parallel changes in gene expression underlying divergent trophic specialization. But on closer inspection, it looks like these parallel changes likely influence parallel adaptations to higher trophic levels presented by these two novel niches. And then this last point, these unique patterns of expression divergence that were unique to each of the specialists seem to be more important for the more obvious phenotypic differences that we saw between species, like craniofacial development and pigmentation. And these are the types of genes that I want to focus on for the last part of my talk, only I want to think about them in the context of hybrids and how selection might shape their expression patterns. So ecological speciation theory suggests that divergent ecological selection can favor divergent adaptive phenotypes, and that reproductive isolation evolves between populations as a byproduct of adaptation. We can also think about this at the level of gene expression, where we can have cis and trans regulatory factors binding to regulate the expression level of some gene. If we have two populations diverging and adapting to different niches, over time, divergent selection may favor regulatory factors that cause low expression of a gene in one species and high expression of the same gene in another species. When these species come back together and hybridize, these co-adapted regulatory alleles will become decoupled, potentially resulting in expression levels that are much lower or much higher in the F1s than either parental population. Now, this type of expression pattern is known as hybrid gene misregulation, which could be caused by Dobzhansky-Muller incompatibilities between divergent regulatory alleles. So now I can rephrase the question I'm asking for this section of my talk is, can divergent ecological selection drive hybrid gene misregulation? If it can, then we should expect to find genes that are important for shaping adaptive phenotypes that showed differential expression between species and misregulation in their F1. Now, this question I'm asking isn't new, but it remains unresolved. Most of what we know about hybrid gene misregulation comes from studies in distantly related species and subspecies of Drosophila and mouse, where in these systems, misregulation has been associated with strong intrinsic reproductive isolating barriers like hybrid sterility and inviability. However, it's debated whether misregulation is the cause or consequence of reproductive isolation in these systems. Furthermore, misregulation in these systems is thought to evolve due to stabilizing selection in allopatry, not divergent selection in sympatry. And that's why I want to look at hybrid gene misregulation in this system. So to do this, I generated 124 new transcriptomes. So forget about all the old gene expression data I was talking about. These are newly generated just for this project. And these were generated from early developing individuals sampled from San Salvador Island, so the sympatric species, including the specialists, as well as outgroup generalist populations. One population from New Providence Island, which is about 300 kilometers away from San Salvador Island, and one population from the coast of North Carolina, which is about 1,000 kilometers away from San Salvador Island. So first I wanted to look for patterns of differential expression between species because I had preliminary data to show that there are many genes differentially expressed. And so I again generated purebred F1s from crosses within species from the same populations. I collected eggs and let them develop again for 8 days before sampling total body mRNA. And then I also sampled this earlier developmental stage 2 days after fertilization. And this is when cranial neural crest cells are migrating to the pharyngeal arches, which is where a lot of craniofacial magic happens early in development. So with these 2 developmental stages, I again generated read counts and asked, can I identify genes that are differentially expressed between species? And so here I'm showing that between the specialists, the molluscivore versus the scale eaters, I found over 4,000 genes differentially expressed at that 8-day-old stage. And this is a lot of differential expression. If we look at the total proportion of genes differentially expressed out of all genes expressed, that's about 30%. And so I wanted to look here on the x-axis at phylogenetic distance between populations using branch lengths from that tree I just showed. And this is because I wanted to understand how genetic divergence scaled with gene expression divergence. And this is because there's some nice predictions that we have about how gene expression should diverge between closely related populations under drift. We expect that expression divergence will increase with phylogenetic distance until it reaches some boundary where gene expression divergence is constrained by stabilizing selection. So relating these predictions to this system, if we have these 3 populations of allopatric generalists that are all adapted to similar ecological niches. We might then expect that drift is a dominant force acting on gene expression divergence between these species. However, if we expect that selection on gene expression has been important in shaping these divergent specialists, then we might expect that gene expression will diverge more rapidly between these sympatric populations compared to these allopatric populations. So let's look at some other comparisons here. So this is what I showed you, the 30% of genes differentially expressed between specialists. If we look at the comparison between San Salvador Island generalists versus molluscivores and generalists versus scale eaters, there are a lot fewer genes differentially expressed. And then looking at the generalist outgroup populations, it looks like about 10% of genes were differentially expressed between the outgroup generalists and the generalists from San Salvador Island. And what I hope is glaringly obvious right here is how much higher this point is than all the other points. And so this is a really strong indicator that selection on gene expression has been important for adaptation to these divergent niches. So this supports the first part of my prediction here about genes being differentially expressed. Now let's look at expression in the F1 hybrids to see if we find patterns of gene misregulation. So to generate these F1 hybrid transcriptomes, I again set up breeding tanks, but this time crossed species to form F1 hybrids, and then collected eggs and extracted RNA in the same way that I did with the purebred F1s. Only this time I also sequenced the parents of the F1 hybrids so that I could determine cis and trans regulatory mechanisms underlying expression divergence between species. So what I wanted to do with these F1 hybrid transcriptomes was ask, can I find genes that are differentially expressed between the hybrids and between their parental populations? And there was a question as to whether we'd find any misregulation, but we found some, which was pretty significant here in the hybrids generated from crosses between molluscivores and scale eaters. At that 8-day-old stage, about 1,500 genes were misregulated in F1 hybrids. And if we again look at the total number of genes misregulated, that's about 10%. And so I again wanted to ask, does it look like hybrid gene misregulation and this expression pattern is being shaped by selection? And so I again thought that maybe our predictions for gene expression divergence also hold for hybrid gene misregulation, where it will increase with increasing phylogenetic distance. So looking at other comparisons again, here between the San Salvador Island generalist and the molluscivores, there was very low hybrid misregulation, whereas the cross between San Salvador Island generalists and scale eaters was similarly high to the misregulation in the specialist crosses. And then looking at the outgroup hybrids, so in the cross between San Salvador Island generalists and New Providence generalists, I found low misregulation, similar to the molluscivore cross. But then in the cross with North Carolina generalists, I found pretty high misregulation. And this was confusing at first, because if we cover this New Providence Island point, it looks like hybrid gene misregulation is maybe diverging with our expectations under drift, with increasing misregulation steadily with phylogenetic distance. But if you cover the North Carolina point, it looks like these scale eater crosses exhibit higher misregulation than we would expect under drift. And so I wanted to think about this in a different way. So now this is the same data, but now changing the x-axis to geographic distance between populations. And then I use branch lengths from that tree I showed you to do a phylogenetic least squares regression. And I found that hybrid gene misregulation was not explained by geographic distance between populations. So what this meant to us is that this high misregulation that we see in these scale eater crosses is higher than we would expect after correcting for phylogenetic relationships between samples, and potentially suggests that selection on gene regulation was important in shaping these patterns. So now we had evidence for selection acting on gene regulation using this phylogenetic framework. But another important prediction here is that the same genes that are differentially expressed between species should also show misregulation in the F1. And excitingly, I found many genes matching this expression pattern. So here in purple are 320 genes that were differentially expressed between specialists, the molluscivore and scale eater, and also misregulated in their hybrids. And there were more than just these 320 genes that I identified a similar expression pattern in crosses between the San Salvador generalist and each of the specialists. So that in total, there were 750 genes matching this expression pattern. So next I wanted to know, do these genes show signs of selection? So first, as a first pass, I looked to see if these genes were near SNPs fixed between populations, and also whether they were in regions of the genome that showed high between population divergence measured as DXY. And I found that 125 of these genes matched these criteria. And I'm referring to these genes as ecological DMI candidate genes. DMI because they show expression patterns consistent with divergent regulatory alleles causing hybrid misregulation, and ecological because these genes were strikingly enriched for developmental processes related to adaptive traits. So we saw enrichment related to craniofacial development, to skeletal muscle development. Here I'm showing that the specialists tend to have larger adductor mandibulae, which are muscles that are important for the force of jaw closure. We saw enrichment related to nitrogen metabolism. I showed you earlier that the specialists occupy a higher trophic position with more nitrogen-rich diets than the generalists. And then we also saw enrichment related to brain development, which could influence a number of different behavioral differences between species. One of my favorite examples being my lab mate Michelle showed that specialists tend to be much more aggressive than the generalists. So we had this evidence that these genes seem to be influencing adaptive traits, but we can also go beyond gene ontology enrichment analyses to confirm this. So this gene, MPP1, is differentially expressed between the generalist and the scale eater, and is also misregulated in their F1s, which show much lower expression. This gene, mutations in this gene have recently been shown to cause craniofacial deformities in humans and mice, and knockouts in mice cause decreased bone density. This gene shows expression patterns consistent with cis-regulatory divergence, with the scale eater allele always being expressed more highly than the generalist allele in the F1 hybrids. And in short, this is really cool because it suggests that there is some linked nearby genetic variation controlling expression divergence between these two species. And we find 170 SNPs that are fixed between the generalists and the scale eaters within 20 kb of the MPP1 gene region in blue here, and each of these black dots corresponding to a fixed SNP. This region also shows signs of selection with high between population divergence and low within population diversity. It also shows that characteristic shift in the site frequency spectrum associated with a hard selective sweep in the scale eaters. And this region is significantly associated with jaw size variation using that same genome-wide association mapping approach I described earlier. And so this is exciting because it suggests that MPP1 and other candidate genes like it are differentially expressed between species due to their influence on divergent adaptive traits. What then does this mean for the hybrids where these seemingly important genes are misregulated? Well, first let me take a step back and point out a couple of really cool things we know about this system. First, we know that hybrids that show craniofacial phenotypes that are intermediate between parental types show low fitness in the wild, suggesting that extrinsic ecological factors are an important source of reproductive isolation in this system. We also know from recent feeding kinematic data that F1 hybrids differ significantly from their scale eater parents in traits that are essential for scale eating, like peak gape of the jaws and gape angle of the jaws. So putting all of this together, if we have divergent selection acting on regulatory machinery of a gene, such that one species shows low expression, the other species shows high expression, and let's say that this gene influences craniofacial development, and this gene shows misregulation in the F1 hybrids, then perhaps that misregulation is causing weak intrinsic isolation. It's also possible that misregulation causes abnormal development of phenotypes and traits that are important for scale eating or ecological specialization. So this misregulation could affect suboptimal feeding kinematics. And this could lead to extrinsic isolation by reducing hybrid fitness in the wild. And so I have one more piece of evidence that supports this middle link here. And it comes from gene expression data from a cross between generalists and molluscivores. And I sampled RNA from craniofacial tissue dissected from adults, purebred and hybrids at that 20-day-old stage when they're a bit bigger. And I found that in these tissues that develop into these divergent morphologies, we find a lot of misregulation in the hybrids, more so than what we see at the 8-day-old stage. And so together, I've shown you that misregulated genes under selection are associated with these adaptive ecological traits in trophic specialists. But I'm hoping that the broader takeaway here that maybe you think about later is that this evidence supports a compelling genetic mechanism of rapid speciation, where misregulation could be driven by adaptive processes and contribute to reduced hybrid fitness and ultimately reproductive isolation between young species. And that's my thesis research. Thanks so much for listening. This is the part where I'm going to go full YouTube streamer and say, look below if you'd like to check out any of the publications associated with these projects and read more about them. We also are going to do a virtual celebration on Zoom. There's a link for that in the video description. And then I'd like to take questions. So if there's anything that you want to ask that's like fresh in your mind right now, feel free to write it out and leave the comment, and I'll check through them while I go through thanking my funding sources and the people that got me here. So first, I would really like to thank the Society for the Study of Evolution for a Rosemary Grant Award that enabled me to do some field research, as well as the Triangle Center for Evolutionary Medicine for a semester-long research fellowship. And then I'd really like to thank my thesis committee, Daniel Matute, Chris Willett, Brian Langerhans, and my committee chair, Bob Duronio. I feel extremely lucky to have had this group of individuals mentorship over the years, and I'm very grateful for the guidance that they've provided me over these years. And then I'd also like to thank my thesis advisor, Chris Martin. I have enjoyed being in this lab so much, and a lot of that is thanks to Chris. Everything that you've seen today started as a conversation in his office. He's been a fantastic mentor, and I'm really proud to say that I'm his first graduate student. So I say that all these projects started as conversations in his office. Here's an example of one of our brainstorming sessions, these scribbles. And at the beginning of grad school, my research felt kind of like these scribbles, scattered mess of pieces that vaguely resembled science. But over time, with a lot of help from Chris, those scribbles became these figures. And I feel like my research went through a similar transformation throughout grad school as I gained a lot of confidence in my ability to do research, and I now feel really prepared for my next steps as a postdoc. And I feel I owe a lot of that to Chris. So thank you for everything. Your enthusiasm for science and contagious motivation really made grad school a fun and rewarding experience. But Chris doesn't get all the credit. I'd also like to thank my lab mates, Emily and Michelle, for making lab really feel like a home, and also for making field expeditions feel like true adventures. I'd like to thank all my other friends that I've made here in Chapel Hill, other graduate students and people living around here. They provided so much a needed balance to my work life, and we created some truly great memories together. I'm very grateful for that. Quick shout out to my cats, Theo and Zansky, and many more thanks to my family, the people that have had the biggest impact on my life. There is no way I would be here without this wonderful group of people that raised me. And I just want to thank everyone for all of their support as I've gone through all of this, so many years of education. Their love and support has been everything. So thank you so much. And I'd also like to thank my fiance, Kamiko. By far, my most important discovery I made during grad school. not only has she been so supportive as a partner, but I also owe so much to her as for my development as a scientist. Not only helping me with practical things like teaching me how to code in Python, but helping with really big questions like helping me discover what I want to do in life. And I'm really excited to continue planning for the future with her. So I just want to say I'm grateful for all that you do, and maybe thank you the most for being my best friend through all of this. Okay. All right. Now I'll see if technology is going to cooperate, and I'll try to take some questions.